Welcome, curious minds, to the deep dive. Today, we're embarking on an incredible journey into the heart of what truly keeps us safe, healthy, and resilient every single day, often without us even realizing it.
That's right.
We're talking about the body's silent, tireless protectors, that intricate, unbelievable network working constantly, tirelessly to keep you safe. Our topic for today's deep dive is clinical immunology. We're going to peel back the layers of the body's magnificent defense system, exploring its functional anatomy, how it springs into action when threats emerge, and critically, what happens when this perfectly orchestrated system goes awry. Our mission: to break down this incredibly complex field, extracting the crucial insights and surprising facts to give you a shortcut to truly mastering this information. This isn't just a surface scratch. We're going deep. In fact, we're going as deep as we possibly can within the time we have to give you the most comprehensive understanding.
And it's not just a fascinating academic exercise. What's truly fundamental here is that understanding the immune system is crucial, not just for doctors or scientists, but for anyone seeking to be genuinely well-informed about human health.
Exactly.
This deep dive will cover everything from the microscopic cellular battles waged within our bodies to the systemic diseases that arise from immune dysregulation, and even how we can manipulate immunity for therapeutic purposes. It's a field brimming with aha moments, and by the end, you'll have a much clearer, more comprehensive picture of this vital system, making you feel like an elite professional in this domain.
Okay, let's unpack this. Before we dive into the malfunctions, the overreactions, or the deficiencies, we really need a solid understanding of how the immune system is supposed to work. It's an intricate dance of organs, cells, and proteins, all working in concert.
Absolutely fundamental.
Fundamentally, the immune system has evolved to identify and destroy pathogens while minimizing any collateral damage to our own healthy tissues. But it's not just about fighting off invaders. We now understand that it also regulates tissue repair after injury. And, quite critically, when the system becomes dysregulated, it can directly govern responses that lead to autoimmune and autoinflammatory diseases.
That dysregulation is key.
If it's dysfunctional or deficient, the immune response can impact potentially every single organ system in your body. It's truly systemic.
It's an all-encompassing protector.
Yeah.
And to better understand how it works, we broadly categorize its defenses into two major responses. There's the innate immune response, which provides immediate generic protection kicking in within minutes.
Like the first responders.
Exactly. Think of it as your body's rapid response, non-specific police force. Then you have the adaptive or acquired immune response, which takes a bit longer to develop, but offers exquisite specificity and long-lasting protection, what we call memory.
The specialists.
Precisely. This is like your body's highly trained specialized forces that remember specific threats. What's absolutely key to remember is that these two systems, while distinct in their approach, are intricately linked and constantly communicating, forming a seamless defense strategically placed throughout the body within lymphoid organs, cells, and soluble proteins.
That makes sense, a first line of defense and then a more tailored long-term approach. So let's start with the innate side, which is your body's first responder, always on patrol. How does that immediate protection manifest?
The innate immune system provides immediate protection primarily by recognizing generic microbial structures, what we call pathogen-associated molecular patterns, or PMPs.
PMPs, right.
These are molecular motifs found on microbes, but not on our own mammalian cells. It's truly fascinating how this system can mobilize within minutes, eliciting essentially identical responses in different individuals, unlike the highly individualized adaptive system.
So everyone's innate response is pretty similar initially.
Broadly speaking, yes. And our physical barriers form the absolute first line of defense. Think of your skin as a multi-layered fortress. Its keratinized cells are tightly packed, physically limiting microbial colonization.
Just a tough wall.
Well, more than that. Beyond being a physical wall, it creates an acidic, microbially unfriendly environment through hydrophobic oils from sebaceous glands, and even produces its own germ killers in sweat.
Sweat kills germs.
It contains things like lysozyme, which destroys bacterial cell walls, and antimicrobial peptides called defensins. Then we have mucous membranes found in your respiratory, GI, and genitourinary tracts.
Right, the moist surfaces.
They secrete mucus, which physically traps invading pathogens. Immunoglobulin A, or IgA, though it actually comes from the adaptive system, interestingly plays a crucial role here by preventing bacteria and viruses from attaching to and penetrating epithelial cells.
Ah, so a bit of adaptive help even at the barriers.
Exactly. There's constant crosstalk. Like the skin, These membranes also employ lysozyme, other antimicrobial peptides, and lactoferrin, which starves invading bacteria by binding essential iron.
Clever, depriving them of resources.
Precisely. In your respiratory tract, cilia trap pathogens and help remove mucus, a process aided by actions like sneezing and coughing.
Expulsion mechanisms.
And in your gastrointestinal tract, strong hydrochloric acid and salivary amylase chemically destroy bacteria, while normal peristalsis and induced vomiting or diarrhea assist in clearing harmful organisms. It's a sophisticated, multi-pronged physical and chemical defense.
So a lot more than just a simple wall.
Yeah.
What about the internal friendly forces that are always there, even before an invader shows up? The good bacteria.
Precisely. We can't overlook the microbiome. Approximately 10-14 commensal bacteria normally reside at our epithelial surfaces in a truly symbiotic relationship.
10 to the 14th, that's a lot.
An enormous number. This endogenous flora provides a constitutive defense. They directly compete with pathogenic microorganisms for resources like space and nutrients, and they produce fatty acids and bactericidins that actively inhibit pathogen growth.
So they fight for turf.
They do. It's a fascinating collaboration, as recent research has even shown that these commensal bacteria help shape our immune response by inducing specific regulatory T cells within the intestine.
Wow, so they actively train our immune system.
In a way, yes. A striking clinical example of their importance is how broad-spectrum antibiotics, which eradicate this normal flora, commonly result in opportunistic infections by organisms like Clostridium difficile, which then rapidly colonize this newly undefended ecological niche.
Right, I've heard about C. diff infections after antibiotics. That makes perfect sense now.
It's a classic example of disrupting that balance.
So if these external and constitutive defenses are breached, then the more active microscopic soldiers come into play. What are the key cellular components of this innate system?
That's right. If the initial barriers are overcome, then the specific soluble proteins and cells of the innate immune system are activated. Let's delve into the cellular components. Phagocytes, literally eating cells, are specialized cells like neutrophils, monocytes, and macrophages. Their core role is to ingest and kill microorganisms, scavenge cellular debris, and produce inflammatory molecules. They're particularly important for defense against bacterial and fungal infections.
The cleanup crew and the warriors combined.
Essentially, yes. Phagocytes are incredibly smart in how they recognize threats. They express pattern recognition receptors, PRRs, on their surface, which bind to those pathogen-associated molecular patterns, PMPs we mentioned earlier, those distinct molecular motifs found on microbes, but not on our mammalian cells.
So they recognize general danger signals from microbes.
Exactly. Examples include receptors that recognize bacterial cell wall components, bacterial DNA, and viral RNA. While PRRs allow direct recognition, engulfment is greatly enhanced by opsonization.
Opsonization sounds important.
It is. It's a process where soluble molecules like C-reactive protein, complement, or even antibodies from the adaptive system act as a bridge for phagocyte receptors, essentially tagging the pathogen for destruction. Makes them easier to grab.
Like putting handles on the bad guys.
A good analogy. This leads to efficient intracellular pathogen destruction and activation of pro-inflammatory genes, which then generate signaling proteins called cytokines.
And among those phagocytes, neutrophils seem to be everywhere, often rising dramatically during an infection. What makes them so special?
Neutrophils are indeed remarkable. Also known as polymorphonuclear leukocytes, these are bone marrow-derived cells that circulate freely. They're short-lived, with a half-life of only about 6 hours.
6 hours? That's incredibly short.
It is, but your body produces an astonishing 10-11 daily, indicating their constant demand. Their functions are to kill microbes, facilitate rapid tissue transit, and non-specifically amplify the immune response.
Quantity over quality, in a way. Or maybe just rapid turnover.
Rapid turnover and deployment. They contain granules, essentially tiny biochemical weapons filled with enzymes and other antimicrobial substances. These granules are deployed to destroy invaders, sometimes even releasing their contents extracellularly.
Like tiny grenades.
You could say that. Neutrophils are rapidly recruited to infection sites by chemical signals called cytokines and chemotatic agents, along with adhesion molecules on blood vessel walls, allowing them to quickly exit the bloodstream and reach the site of battle. The rise in neutrophil count during early infection reflects this mass mobilization.
That spike in the white blood cell count.
Exactly. Once in infected tissue, they engulf microorganisms into specialized compartments called phagolysosomes, where killing occurs through both oxidative mechanisms, often called the respiratory burst. a highly effective process using reactive oxygen species and non-oxidative mechanisms involving their potent bactericidal enzymes.
Respiratory burst. Sounds intense.
It is a very powerful killing mechanism. A fascinating, relatively recently identified form of killing is neutrophil extracellular trap net formation, or netosis. Activated neutrophils can actually release a sticky web of chromatin and granule proteins to immobilize and kill microbes without even phagocytosing them.
They throw out a net made of their own DNA.
Precisely. Essentially sacrificing themselves in the process, which can lead to pus and abscess formation as the cellular debris accumulates.
Incredible. So if neutrophils are the rapid response, short-lived shock troops, what about monocytes and macrophages? Are they more like the long-term strategic forces?
Exactly. Monocytes are precursors to tissue macrophages. They're produced in the bone marrow, circulate briefly in the blood, and then differentiate in tissues where they can reside for long periods.
So they settle down in different organs.
Right. Specialized tissue macrophages include Kupffer cells in the liver, alveolar macrophages in the lung, and microglial cells in the brain, each adapted to their specific tissue environment. Macrophages not only perform phagocytosis and microbial killing, but crucially, they also play a vital role in amplifying and regulating the inflammatory response by producing cytokines that drive systemic effects like fever and acute phase protein production, and that stimulate further immune cell recruitment.
So they're directors as well as soldiers?
Very much so. They're constantly surveying their surroundings for tissue damage or invading organisms, engaging in tissue surveillance. They're also essential for clearance, resolution, and repair, scavenging necrotic cells, clearing toxins, promoting tissue remodeling, and aiding wound healing.
The cleanup after the battle, too.
And importantly, macrophages form a direct link between the innate and adaptive immune systems by presenting antigens to T cells and having their activity boosted by T cell-derived cytokines in a powerful positive feedback loop.
Ah, another connection point. Very integrated.
Highly integrated.
And dendritic cells, what's their role in this complex web? They sound like central communicators.
They absolutely are. Dendritic cells are specialized antigen-presenting cells, APCs, found primarily in external contact tissues like skin and mucosa. Their job is to sample the environment for foreign particles.
Sentinels.
Exactly. Once they sense a threat, they activate and then carry microbial antigens to regional lymph nodes where they act as crucial communicators interacting with T and B cells to initiate and shape the adaptive immune response. They are truly the bridge between the immediate innate response and the tailored adaptive response.
The messengers carrying vital intelligence. Got it. And what about these natural killer cells? They sound pretty formidable.
They certainly are. Natural killer NK cells are a unique type of large granular lymphocyte that plays a major role in defense against tumors and viruses.
So cancer and viruses, big targets.
Major targets. While they have features of lymphocytes, crucially, they are not antigen-specific and do not generate immunological memory. They're part of the innate system, ready to act immediately.
Innate, but lymphocyte-like. Interesting.
Yes, NK cells express both stimulatory and inhibitory receptors. Inhibitory receptors typically recognize healthy self-markers, specifically HLA molecules expressed on normal nucleated cells, preventing NK attack.
So they check for an ID badge on normal cells.
Essentially, yes. However, if cells become infected by viruses or undergo malignant change, they can downregulate these HLA molecules. This normally helps them evade adaptive T lymphocyte responses, but it simultaneously abrogates the inhibitory signals to NK cells, leading to NK attack on the abnormal target cell.
Ah, so if the ID badge is missing or altered, the NK cell attacks. Brilliant.
It's a brilliant system of checks and balances. NK cells can also be activated by binding to antigen-antibody complexes, a process known as antibody-dependent cellular cytotoxicity, ADCC, which is analogous to the opsonization we discussed earlier.
So antibodies can guide them too.
They can. Once activated, NK cells kill their targets by secreting perforin, which forms pores in the target cell membrane, and granzymes, which are proteolytic enzymes that cause the target cell to undergo programmed cell death or apoptosis.
A lethal injection, basically.
A very targeted one. They also produce cytokines like TNF-alpha and IFN-gamma, which have direct antiviral and anti-tumor effects.
So they kill directly and also call for backup.
Correct. They're multifaceted killers.
And finally, for the cellular components of the innate system, what about mast cells and basophils? We often hear about them in the context of allergies.
You're right to connect them to allergies. Mast cells and basophils are bone marrow-derived cells central to allergic disorders. Mast cells primarily reside in tissues exposed to the external environment, like skin and gut, providing a crucial sentinel role.
Stationed at the borders.
Right. Basophils, on the other hand, circulate in peripheral blood and are recruited to tissues during inflammation. Both types of cells contain large cytoplasmic granules filled with vasoactive substances, notably histamine.
Histamine, the allergy molecule.
One of the main ones, yes. They express specific receptors on their cell surface that bind IgE antibodies. When a specific antigen or allergen is encountered and it cross-links these Ig-coated mast cells, it triggers the rapid release of histamine and other preformed mediators from their granules and stimulates the synthesis of additional mediators like leukotrienes, prostaglandins, and cytokines.
A rapid degranulation.
Very rapid. This initiates a rapid inflammatory cascade, increasing local blood flow and vascular permeability, stimulating smooth muscle contraction, and increasing mucus secretion and mucosal surfaces. All those familiar symptoms of an allergic reaction.
Sneezing, itching, swelling. Yeah, okay. So beyond the cells, there's a whole network of signaling molecules, like cytokines. These are the internal messages that coordinate the immune response, correct?
Exactly. They are the communication network. Cytokines are signaling proteins produced by immune cells, and many other cell types, over 100 have been identified, each with specific roles.
A huge family of signals.
A very large and complex family. They have complex, overlapping roles in cellular communication and immune response regulation. Subtle differences in cytokine production, especially early in an immune response, can significantly impact the outcome, essentially determining the battle plan.
So the initial signals really matter.
Critically, cytokines bind to specific receptors on target cells, activating downstream intracellular signaling pathways. Think of these pathways as internal messaging systems or master switches within cells that lead to changes in gene transcription and cellular function.
Turning genes on and off.
Precisely. For instance, the NF-POO pathway and the JEC-STAT pathway are two crucial ones. These pathways are central to turning on and off critical immune responses. Understanding them is profoundly clinically relevant. Cytokine inhibitors, often monoclonal antibodies, and small molecule inhibitors that target these pathways.
Like JA inhibitors.
Exactly, like tofacitinib or baricitinib, are now routinely used to precisely control inflammation in autoimmune diseases and cancers, offering truly targeted therapies.
That's a huge breakthrough, knowing how to turn these master switches on and off therapeutically. What about the complement system? That sounds like something out of a sci-fi movie.
It's certainly a powerful force. The complement system is another critical soluble component, consisting of over 20 tightly regulated, functionally linked proteins produced primarily in the liver, circulating in inactive form.
Inactive until needed.
Correct. This system promotes inflammation and eliminates pathogens, acting like a rapidly amplified biological cascade, very similar to how blood clots form. It can be activated by three main pathways. The alternate pathway, triggered directly by microbial surfaces. The classical pathway, usually initiated when antibodies bind to an antigen. And the lectin pathway, which recognizes specific sugar patterns on microbial surfaces.
3 ways to kick it off.
Yes, providing redundancy and specificity. All 3 pathways converge to activate a central protein called C3, which then leads to the assembly of the membrane attack complex, MAC.
Membrane attack complex. Definitely sounds sci-fi.
It's quite descriptive. The MAC literally punctures the cell wall of target cells, causing osmotic lysis. They pop, essentially. This MAC is particularly important in defense against encapsulated bacteria like Neisseria species.
Ah, like meningitis bacteria.
Exactly. Beyond direct killing, complement fragments also act as opsonins, enhancing phagocytosis, and as chemotactic agents, actively drawing other immune cells to inflammation sites. They also link to the adaptive system by targeting immune complexes to antigen-presenting cells, and they help resolve inflammation by dissolving the very immune complexes that triggered them, minimizing bystander damage.
So it kills, tags, attracts, and cleans up. Very versatile.
Extremely versatile. A prime clinical example of its importance is the monoclonal antibody eculizumab, which targets a component of complement and is used for specific rare blood disorders. However, inhibiting this system highlights the risk of invasive infections, particularly meningococcal sepsis, when this protective mechanism is dampened.
A real trade-off there. So all these parts of the innate system, the physical barriers, the cells, the soluble molecules, are constantly interacting. What's the final piece of the puzzle here regarding these soluble molecules?
The last crucial soluble players we'll mention are integrins. These are transmembrane proteins that are absolutely crucial for cell-cell and cell matrix interactions.
Sticking cells together and to their surroundings?
Yes, they mediate cellular attachment, signal transduction, and critically cell migration. Their role in autoimmune disease has been extensively studied, leading to targeted therapies. For instance, natalizumab, an antibody that blocks a specific integrin, is used in multiple sclerosis to prevent immune cells from entering the central nervous system, effectively preventing them from causing damage in the brain.
Blocking their entry pass, essentially.
A good way to put it.
So, the innate system is our rapid response team, a complex orchestra of cells and molecules, always ready to spring into action. It's truly amazing how quickly and effectively it mobilizes to contain a threat. But what happens when that initial wave isn't enough, or when the threat requires a more tailored, long-term approach? This is where the immune system needs to learn, right?
That's precisely where the adaptive system comes in. This raises an important question about how the immune system not only adapts, but also remembers specific threats. That's where the adaptive system truly shines, with its incredible precision and memory providing that bespoke, long-lasting defense.
Okay, the adaptive system. What makes it so different?
The adaptive immune system is mobilized if the innate system doesn't provide effective protection. It has three truly remarkable characteristics. First, exquisite specificity. It can discriminate minute differences in molecular structure, making its targeting incredibly precise.
Really fine-tuned recognition.
Extremely. Second, it's highly adaptive, meaning it can respond to an almost unlimited number of different molecules, even ones it has never encountered before. And third, it possesses immunological memory, allowing a much more effective and rapid response on subsequent encounters with the same antigen.
Specificity, adaptability, and memory. Got it.
There are two major arms to this system. Humoral immunity, which primarily involves B lymphocytes producing antibodies, and cellular immunity, which involves T lymphocytes that synthesize cytokines and directly kill target cells. These two arms interact closely with each other and are in constant dialogue with the innate components we just discussed.
Humoral and cellular. And where does all this adaptive action take place? Are there specific headquarters or training grounds for these cells?
Absolutely. The lymphoid organs are central to this entire operation. We divide them into two categories. Primary lymphoid organs are where lymphocytes develop and mature.
The schools, basically.
Yes. The bone marrow is where hematopoietic stem cells originate and differentiate into T and B lymphocytes. And it's also where B lymphocytes complete their maturation. The thymus is the specific site of T-cell maturation, a bilobed structure located in your chest.
The T-cell university.
A fitting analogy. It's most active in fetal and neonatal periods and gradually involutes after puberty. While failure of thymic development leads to profound T-cell deficiency, surgical removal in childhood generally isn't associated with significant immune dysfunction, as most T-cell development has already occurred.
So timing matters for thymic function.
Very much so. Then we have secondary lymphoid organs, which are essentially the meeting points where mature lymphocytes encounter antigens and get activated.
The battlegrounds or communication hubs.
Both, really. The spleen is the largest secondary lymphoid organ. It filters blood and performs vital phagocytosis of senescent red blood cells, bacteria, and immune complexes. It's also an important site of antibody synthesis and is crucial for defense against encapsulated bacteria, making individuals without a spleen vulnerable to infections from organisms like Streptococcus pneumonia and Haemophilus influenza.
Right. Spleen removal increases infection risk.
Significantly, especially for those encapsulated bacteria. Lymph nodes are strategically positioned throughout the body, maximizing exposure to lymph draining from external contact sites. They are highly organized structures containing specialized areas for B lymphocytes, T lymphocytes, and antigen-presenting cells.
It's like little garrisons filtering the lymph fluid.
That's a good way to think of it. And finally, mucosa-associated lymphoid tissue, MALT, consists of lymphoid cells diffusely distributed along mucosal surfaces, functionally similar to lymph nodes, including structures like tonsils, adenoids, and Peyer's patches in the gut. All these lymphoid tissues are connected by a network of lymphatics, which not only provide access to lymph nodes, but also return interstitial fluid to the venous system and transport fats.
So a highly organized system with specific training grounds and meeting points. Let's delve deeper into humoral immunity. What are B lymphocytes and antibodies, and how do they work their magic?
Excellent question. Humoral immunity is primarily mediated by B lymphocytes and antibodies. B lymphocytes differentiate from hematopoietic stem cells in the bone marrow. Their primary functions are antibody production, interaction with T cells, and antigen presentation.
Antibody factories and communicators.
Yes. You'll find mature B cells in bone marrow, lymphoid tissue, the spleen, and circulating in the blood. Each B cell expresses a unique B cell receptor, which is essentially an immunoglobulin on its surface. perfectly designed to bind specific soluble antigen targets.
A unique key for a specific lock.
Precisely. When a B cell encounters its specific antigen, typically in a lymph node, and receives the right cytokines and signals from T helper cells, it undergoes rapid clonal expansion.
Makes lots of copies of itself.
Exactly. It makes many copies of itself, all specific to that antigen. This process is accompanied by somatic hypermutation, a clever genetic fine-tuning that generates B cells with even higher affinity receptors for the antigen.
They get better at grabbing the target.
Yes, affinity maturation. These highly adapted cells then differentiate into either long-lived memory cells, which reside in lymph nodes and are ready for future encounters, or plasma cells, which are antibody factories, churning out millions of antibodies. Those memory cells are the reason your body can respond faster and more effectively upon subsequent exposure to the same pathogen.
And these antibodies, or immunoglobulins, or what we hear about when we talk about immunity, what exactly are they and how do they function?
Immunoglobulins, Ig, or antibodies, are soluble proteins produced by those plasma cells. Structurally, each antibody is composed of 2 heavy chains and 2 light chains. The heavy chain determines the antibody class or isotype. We have IgG, IgA, IgM, IgE, and IgD, each with specific subclasses.
Different types for different jobs.
Exactly. The variable regions, known as the Fab regions, are what recognize and bind to antigens. The constant region, or FSE region, dictates the antibody's functional consequences, such as how it interacts with other immune cells.
So one part binds the target, the other part signals what to do.
You've got it. Antibodies have several crucial functions. They act as opsonins, essentially tagging pathogens for destruction and greatly facilitating phagocytosis. They also facilitate cell killing by cytotoxic cells, particularly NK cells via ADCC. IgM and IgG are potent activators of the classical complement pathway, linking directly to the innate system we discussed.
Tagging, helping NK cells kill, activating complement.
And importantly, antibodies can directly neutralize antigen biological activity, preventing pathogens from causing harm. IgA is especially important for this at mucosal surfaces.
So they can just block the pathogen's function directly?
Sometimes, yes. Just to quickly go over the classes and their main properties. IgG is the most abundant antibody, found equally in blood and extracellular fluid. It's the only antibody that can cross the placenta, providing crucial passive immunity to newborns.
The main workhorse, and protects the baby.
Right. IgA is vital at mucosal surfaces, like in your gut and respiratory tract, and is highly effective at neutralizing toxins.
Guardian of the gates.
IgM is the first antibody produced in a primary immune response, and is particularly effective at agglutinating or clumping pathogens.
The first responder antibody, good at grabbing multiple targets at once.
IgE is central to allergic disease and plays a role in defense against parasitic infections, primarily by binding to mast cells, basophils, and eosinophils.
The allergy and parasite specialist.
And IgD has a role in B cell development, though its exact functions are still being fully elucidated.
Still a bit mysterious. So antibodies are essentially our body's highly specific smart bombs. This concept of immunological memory seems to be central to how vaccines work. Can you explain the difference between a primary and secondary response?
It's absolutely foundational to vaccination. In a primary response, when a naive B cell is activated by an antigen for the first time, IgM antibodies are produced first, typically within 5-10 days.
IgM comes first.
Yes. Other classes like IgG, IgA, and IgE follow after 1-2 weeks, and their production is T-cell dependent. However, in a secondary response, when a memory B cell is re-exposed to the same antigen, the lag time for antibody production is dramatically shorter, usually only 2-3 days.
Much faster.
Much faster. The amount of antibody produced is much greater, and is dominated by high-affinity IgG. Crucially, this rapid and robust secondary response does not require additional T lymphocyte input, making it incredibly efficient. This rapid, specific, and strong response is precisely what makes vaccines so incredibly efficacious, priming your immune system to quickly defeat future encounters with a pathogen.
Fascinating. That difference is huge. So if humoral immunity is about antibodies, what about cellular immunity? What role do T lymphocytes play, and how do they identify their targets?
Cellular immunity, mediated by T lymphocytes, is vital for defense against viruses, fungi, and intracellular bacteria. They also play a critical immunoregulatory role, essentially orchestrating and regulating other immune components.
The conductors of the immune orchestra.
A very apt description. T lymphocyte precursors develop in the bone marrow, but uniquely they must mature in the thymus. Each T cell has a unique, highly specific T cell receptor, TCR. In the thymus, a stringent selection process occurs, eliminating autoreactive cells that might attack your own tissues, thereby ensuring self-tolerance.
Quality control to prevent friendly fire.
Extremely important quality control. Mature T cells then populate other immune organs. Your body has an estimated 107 to 109 T cell clones, ensuring broad antigen recognition capabilities.
An immense repertoire.
Now, a key difference from B cells is that T cells cannot recognize intact protein antigens floating freely. Instead, antigen-presenting cells, APCs, like the dendritic cells and macrophages we discussed, and even B cells break down proteins into smaller peptides and then present them in association with HLA molecules on their surface.
So the antigen has to be processed and presented on a silver platter, so to speak.
Exactly. The T cell then recognizes this specific peptide HLA complex, a crucial handshake. The extensive polymorphism of HLA molecules among individuals ensures immense diversity in antigen recognition across the population.
That's why tissue matching is so important for transplants, right? The HLA molecules.
Precisely. While all nucleated cells can process and present antigens, specialized APCs possess co-stimulatory molecules that provide a necessary second signal for full T cell activation, acting as an extra layer of security.
A two-factor authentication for T cell activation.
Yes, you can think of it that way. Prevents accidental activation.
So T cells are like highly specialized scouts, able to differentiate between self and non-self, but only when presented properly. Are there different types of T cells, each with their own mission?
Yes, absolutely. T lymphocytes are divided into two main subgroups based on their CD, cluster of differentiated antigen expression, and the type of HLA molecule they recognize. CD8 plus T lymphocytes. These recognize antigenic peptides presented with HLA class I molecules. Their primary function is to directly kill infected cells.
The killer T cells.
Yes, sometimes called cytotoxic T lymphocytes or CTLs. They kill by producing pore-forming molecules like perforin and proteolytic enzymes called granzymes, which trigger apoptosis. They're particularly important in defense against viral infections and cancer. CD4 plus T lymphocytes, also known as T helper cells. These recognize peptides presented on HLA class II molecules. Their role is mainly immunoregulatory.
The helper cells, they don't kill directly.
Generally not their main role. They produce cytokines and provide crucial co-stimulatory signals that support both CD8 plus T cells and B cells. They also interact closely with phagocytes, influencing cytokine production.
So they help everyone else do their jobs better.
Essentially, yes. And CD4 plus cells are further subdivided by their cytokine production and roles. Th1 cells are pro-inflammatory and support crucial delayed type hypersensitivity reactions. Th2 cells promote allergic responses and activate B cells and eosinophils.
Th1 versus Th2. Key balance, I imagine.
A very important balance, often skewed in certain diseases. T regulatory cells, Tregs, are a specialized subset that are anti-inflammatory, actively suppressing other immune cells and playing a vital role in preventing autoimmune disease.
The peacekeepers.
You could say that. Essential for maintaining tolerance. And Th17 cells are pro-inflammatory, involved in mucosal immunity, and help control extracellular pathogens, often synergizing with other inflammatory cytokines.
Another pro-inflammatory player. That's a lot of different T cell players. How is this incredible T cell activation regulated to prevent them from causing collateral damage or attacking our own cells?
This is a very tightly regulated process, a constant balance between activation and inhibition. T-cell activation regulation relies on a delicate balance of co-stimulatory molecules, which are like the accelerators of the immune response.
The gas pedal.
Right, and inhibitory molecules, which act as the brakes.
Got it. Accelerators and brakes.
For example, CD28 binding to other molecules provides a co-stimulatory signal, as does the T-cell's own production of IL-2. But inhibitory molecules, such as CTLA4, which competes with CD28 for binding, and PD-1 binding to PD-L1, provide crucial inhibitory signals, preventing overactivation.
CTLA4 and PD-1. Those sound familiar from cancer therapy.
Exactly. This regulatory balance is clinically highly significant. Drugs like abatacept, which is a CTLA4 Fc fusion protein, are used therapeutically to inhibit T-cell activation in conditions like rheumatoid arthritis and solid organ transplantation, showcasing how we can manipulate these pathways to treat disease, and checkpoint inhibitors in cancer work by blocking these brakes.
So the immune system is always on patrol, identifying threats. That constant vigilance and precision are incredible. But when those defenses are breached, we see something everyone recognizes: inflammation. It's often misunderstood, sometimes seen as purely negative, but it's absolutely crucial for healing. By definition, inflammation is the tissue's fundamental response to injury or infection, and it is necessary for normal repair and healing to occur.
You're absolutely right. It's a double-edged sword. This is where the innate system really shines, mobilizing rapidly to contain a threat. But if uncontrolled, it can indeed cause significant damage.
So let's talk about that immediate acute response.
Let's discuss acute inflammation. It's a rapid and complex interplay between the cells and soluble molecules of the innate immune system. The classical external signs familiar to us all include heat, redness, pain, and swelling, the very visible hallmarks of a battle being fought.
Kaler, rubor, dolor, tumour, the old Latin terms.
Exactly. This inflammatory process is immediately initiated by local tissue injury or infection. Early events involve damaged epithelial cells producing cytokines and antimicrobial peptides, which quickly lead to an infiltration of phagocytic cells like neutrophils. Key mediators released within the inflamed tissue, such as leukotrienes, prostaglandins, histamine, and others, cause vasodilation, increasing blood flow, and dramatically increasing vascular permeability.
Making the blood vessels leaky.
Precisely. This allows vital fluid, proteins, and immune cells to rapidly traffic into the affected tissue, essentially flooding the battleground with defenders. Additionally, potent pro-inflammatory cytokines like IL-1, TNF-alpha, and IL-6, produced at the injury site, are released systemically.
So the alarm spreads beyond the local site.
It does. These act on the hypothalamus to cause fever, signaling a body-wide emergency, and on the liver to stimulate the production of acute phase proteins.
Ah, the acute phase response. We hear a lot about C-reactive protein, or CRP, in that context. What exactly is the acute phase response, and what are those proteins doing?
The acute phase response refers to the liver's rapid production of various proteins in response to inflammatory stimuli. These proteins have a wide range of activities aimed at containing the threat. C-reactive protein, CRP for example, is a superstar in this response and can increase up to 1,000-fold.
1,000-fold increase. Wow.
It's dramatic. It acts as an opsonin, helping to tag pathogens for destruction, and is a direct biomarker of acute inflammation, rising within 6 hours and having an 18-hour half-life. This means its levels fall promptly once the inflammatory stimulus is removed, making sequential CRP measurements incredibly useful for monitoring disease activity and response to treatment.
So it tells you if inflammation is active right now.
Pretty much, yes. It's generally preferred over the erythrocyte sedimentation rate, ESR, because it's a more sensitive and quicker early indicator. Although there are exceptions where CRP might not rise much despite active inflammation, like an SLE or ulcerative colitis, a high CRP in those cases might actually suggest an infection on top of the underlying disease.
Interesting nuances there. What other proteins are involved?
Other crucial acute phase proteins include serum amyloid A, also increasing dramatically, and fibrinogen, which is essential for wound healing and also contributes to ESR. We also see proteins like alpha-1 antitrypsin that control the pro-inflammatory cascade by neutralizing destructive enzymes, preventing widespread tissue damage.
Putting the brakes on the destructive enzymes.
Right. Antioxidants like haptoglobin scavenge harmful oxygen-free radicals, and iron-binding proteins like ferritin and lactoferrin decrease iron availability for bacteria, essentially starving the invaders. It's important to remember that immunoglobulins are not acute phase proteins, though they may increase in chronic inflammation.
So a lot of different proteins, each playing a specific role in containing the battle. How do doctors measure this in the lab? What are the key lab features of inflammation?
When we look at laboratory features of inflammation, we often see leukocytosis, which is an increase in white blood cells, reflecting the transit and activation of neutrophils and monocytes. Platelet counts can also increase. Chronic inflammation frequently presents with a normocytic normochromic anemia, basically, normal-sized red cells, but fewer of them.
Anemia of chronic disease.
Exactly. Now, the erythrocyte sedimentation rate, ESR, is an indirect measure of inflammation, quantifying how fast red blood cells fall through plasma. This is determined by the plasma protein composition and red blood cell morphology.
How fast the red cells settle in a tube?
Correct. Normally, red blood cells are negatively charged and repel each other, but positively charged plasma proteins, especially acute phase proteins like CRP and fibrinogen, neutralize this charge, causing red cells to aggregate into rouleau stack of coins-like formations. These rouleaux have a higher mass to surface area ratio and thus sediment faster.
So more clumping means faster falling?
Precisely. So, while an acute phase response is a common reason for increased ESR, other conditions not affecting acute phase proteins, such as those causing increased immunoglobulins like multiple myeloma, can also raise it. Conversely, abnormal red cell morphology, as in spherocytosis or sickle cell anemia, can prevent rouleau formation, leading to an inappropriately low ESR.
So ESR isn't always straightforward.
It needs interpretation. This is why, if there's a discrepancy between ESR and CRP, one must assess the individual determinants of ESR. Plasma viscosity is another useful surrogate measure of plasma protein concentration. Like ESR, it's affected by large plasma proteins, but unlike ESR, it's not affected by erythrocyte properties and is generally considered a more reliable and direct inflammation marker.
Plasma viscosity. Less common, but maybe more reliable.
In many situations, yes.
So inflammation is a necessary evil, but it can also go completely out of control, leading to something as devastating as septic shock. What happens when the immune system goes into such overdrive?
Septic shock is indeed an extreme manifestation of overwhelming inflammation. It's characterized by excessive pro-inflammatory cytokine production by macrophages, often triggered by severe bacterial infections, like gram-negative bacteria releasing lipopolysaccharide.
A massive cytokine storm.
Exactly. The consequences are dire. Widespread vasodilation leads to profound hypotension. Fluid leaks from vessels causing hypovolemia and tissue edema. Uncontrolled neutrophil activation releases proteases and oxygen-free radicals within blood vessels, severely damaging the vascular endothelium and further increasing capillary permeability.
Widespread vascular damage.
Yes, this, combined with direct activation of the coagulation pathway, leads to clot formation within damaged vessels, disseminated intravascular coagulation, or DIC. Clinically, this can result in cardiovascular collapse, acute respiratory distress syndrome, ARDS, affecting the lungs, DIC, multi-organ failure, and tragically, often death. Early recognition and intervention are absolutely critical in such cases.
That sounds like a body-wide catastrophe.
Yeah.
So the acute response is a fiery, vital battle, but it must resolve. It's designed to be a temporary, targeted event. What happens when it doesn't and inflammation becomes a prolonged issue, simmering beneath the surface?
If the inflammatory stimulus isn't cleared or controlled, the acute response can unfortunately morph into something more persistent, leading to chronic inflammation and significant collateral damage.
A fire keeps smoldering.
Precisely. Chronic inflammation occurs when the immune response fails to effectively clear or control the initial stimulus. This manifests as a persistent accumulation of neutrophils, macrophages, and activated T lymphocytes within the affected tissues. If this is associated with local deposition of fibrous tissue, a granuloma may form.
Granulomas, like in TB.
Exactly. Granulomas are characteristic of diseases like tuberculosis and leprosy, where the microorganism has a robust cell wall that shields it from being killed despite phagocytosis, leading to a walled-off, persistent inflammatory response. It's a way for the body to contain a threat it can't fully eliminate.
Now that we understand the intricate basics of the immune system and the vital role of inflammation, let's pivot to what happens when things can go seriously wrong. The first big red flag we often see, something that brings patients to their doctors, is recurrent infections.
Indeed. While infections can happen to anyone, recurrent infections, especially if they are frequent, severe, or caused by unusual organisms or at unusual sites, should always raise suspicion of an underlying immune deficiency.
So, frequency and severity are key clues.
Absolutely. Recurrent infections can be attributed to defects in the number or function of specific components. Phagocytes, B cells, T cells, or complement. For instance, T cell deficiencies can lead to infections by pathogens from all groups, bacteria, viruses, fungi, and protozoa, because T cells orchestrate so much of the immune response.
T cell problems affect everything.
Pretty much. In terms of clinical assessment, there are specific warning signs of primary immune deficiency that warrant attention. For children, this might include 4 or more new ear infections or 2 or more serious sinus infections within a year. For adults, it's often 2 or more new ear or sinus infections within a year in the absence of allergy, or 2 or more pneumonias within a year.
Specific numbers to watch out for.
Yes, guidelines exist. Other red flags for both include recurrent deep skin or organ abscesses, persistent thrush, needing intravenous antibiotics to clear infections, or a family history of primary immune deficiency. The presence of 2 or more of these signs may indicate a primary immunodeficiency.
So multiple signs are more concerning.
Definitely warrants investigation. When it comes to investigations, initial steps should include a full blood count with white cell differential, C-reactive protein, CRP, basic organ function tests, a urine dipstick, serum immunoglobulins with protein electrophoresis, and an HIV test.
Starting with the basics.
Always. Further microbiological tests, virology, and imaging help identify the specific organism and infection site. If primary immune deficiency is suspected after these initial findings, more specialized tests are needed.
Digging deeper.
Right. These might include complement functional assays, test vaccinations to assess how well the humoral system responds, specific neutrophil function tests, lymphocyte immunophenotyping to count specific subsets of T, B, and NK cells, lymphocyte proliferation assays to see if immune cells can properly multiply, cytokine production tests, and eventually genetic testing under specialist supervision if a specific primary immune deficiency is suspected.
A whole battery of specialized tests.
It can be quite extensive. For management, if an immune deficiency is suspected but not yet fully characterized, patients should not receive live vaccines due to the risk of vaccine-induced disease. Further management is tailored to the specific underlying cause, often involving aggressive infection treatment and sometimes long-term prophylactic antibiotics.
No live vaccines, that's a crucial point. Beyond recurrent infections, another puzzling symptom that points to immune dysregulation is intermittent fever. This isn't just your run-of-the-mill fever, it suggests something deeper, a systemic issue that comes and goes.
The wide differential diagnosis for intermittent fever includes recurrent infection, malignancy, and certain rheumatic disorders like Still's disease, vasculitis, and systemic lupus erythematosus. However, a familial fever syndrome is a distinct potential cause.
Fevers that run in families.
Essentially, yes. These are genetic disorders resulting from mutations in genes that aberrantly regulate the inflammatory response. The symptoms stem from the activation of intracellular signaling pathways involved in inflammation, leading to the overproduction of pro-inflammatory cytokines, especially IL-1.
So the inflammation switch gets stuck on periodically.
That's a good way to put it. In clinical assessment, a full history and physical examination are vital, paying attention to ethnic background and any family history of similar disorders. If there's no evidence of infection, malignancy, or rheumatic disorder, and there's a positive family history, along with an early age of onset, the likelihood of a familial fever syndrome increases significantly.
Ruling out other causes first.
Absolutely. Investigations involve a full blood count, ESR, CRP, and organ function tests. Serum ferritin should be checked, as very high levels support Still's disease. Blood and urine cultures, and an autoimmune screen including anti-nuclear antibodies, ANA, and antineutrophil cytoplasmic antibodies, ANCA, are also performed. Imaging may be needed to rule out occult infection. If still no other cause is found, genetic analysis should be considered to confirm a familial fever syndrome, though a negative genetic test doesn't entirely exclude one.
Genetics can help, but aren't always the full picture.
Correct. Management begins with NSAIDs for symptomatic relief. If ineffective glucocorticoids can be tried, provided infection is ruled out. If a familial fever syndrome is confirmed, definitive therapy is initiated based on this specific diagnosis, often involving targeted therapies to block the specific cytokines involved.
Targeted treatments based on the specific genetic fault.
That's the goal.
And then there's anaphylaxis, a truly terrifying and rapid immune overreaction that demands immediate attention. Here's where the immune system's normally helpful inflammatory response goes catastrophically wrong, unleashing a systemic crisis that can be life-threatening.
Indeed, anaphylaxis is a powerful and stark example of how quickly the immune system can shift from protective to profoundly damaging. It's about understanding the specific triggers and the cascading release of mediators that lead to such a dramatic systemic response.
It happens so fast.
Incredibly fast. Anaphylaxis is defined as a life-threatening systemic allergic reaction characterized by circulatory collapse, bronchospasm, laryngeal stridor, often accompanied by angioedema and urticaria. The risk of death is increased in patients with poorly controlled asthma and when adrenaline treatment is delayed.
Delaying adrenaline is dangerous.
Critically dangerous. Its etiology involves an allergen binding to and cross-linking membrane-bound IgE on mast cells in a susceptible individual. This triggers the rapid release of histamine, tryptase, and other vasoactive mediators. These mediators cause widespread effects, including vasodilation and increased capillary permeability, leading to dangerous hypotension and bronchoconstriction.
The mast cells basically explode with inflammatory chemicals.
But very rapid degranulation, yes. While historically distinguished as IgE-mediated anaphylaxis versus non-IgE-mediated mast cell degranulation, they are clinically indistinguishable in their life-threatening presentation.
Looks the same, feels the same, just as dangerous.
Exactly. Common triggers for IgE-mediated anaphylaxis include foods like peanuts, tree nuts, fish, shellfish, milk, eggs, and soy products, insect stings, bee, wasp, and certain chemicals or drugs such as intravenous anesthetic agents and penicillin or latex. Non-IgE-mediated triggers can include drugs like aspirin, NSAIDs, opiates, and radiocontrast media, or physical factors like exercise and cold. In about 20% of cases, no cause is identified, termed idiopathic anaphylaxis.
So many potential triggers.
It can be challenging to pinpoint sometimes. The clinical features of anaphylaxis typically include itching, often starting in palms, soles, and genitalia, a profound feeling of impending doom, loss of consciousness, conjunctival injection, flushing, sweating, hypotension, urticaria hives, wheezing, bronchoconstriction, angioedema of the lips and mucous membranes, abdominal pain, diarrhea, cardiac arrhythmias, and potentially life-threatening laryngeal obstruction or stridor.
A whole body reaction.
Truly systemic. It's crucial to recognize mimics of anaphylaxis, which include conditions like vasovagal syncope, cardiac arrhythmias, cardiogenic shock, status asthmaticus, pulmonary embolus, C1 inhibitor deficiency, idiopathic angioedema, and systemic mastocytosis.
Things that can look similar but aren't true anaphylaxis.
Correct, and require different management. In assessment, the severity of the reaction and the time from allergen exposure to symptom onset are key indicators. A detailed 24-hour history helps identify potential triggers. The route of allergen exposure can influence the dominant symptoms. For example, inhaled allergens often cause wheezing. Potentiating factors like exercise or alcohol can lower the threshold for an anaphylactic event.
History is crucial.
Absolutely. For investigations, measuring serum mast cell tryptase is useful to confirm mast cell degranulation, though it can't distinguish between IgE-mediated and non-IgE-mediated reactions. It typically peaks 1 to 2 hours post-reaction. Specific IgE tests can confirm hypersensitivity and are often preferred over skin prick tests after an anaphylactic event.
Tryptase confirms the mast cells were involved.
It's a key marker. The management of an acute event is critical and requires immediate action. First, prevent further allergen contact, ensure airway patency, and prevent hypoxia. The most crucial step is to administer adrenaline, epinephrine, promptly intramuscularly.
Adrenaline first, adrenaline fast.
Absolutely. 0.31.0 milliliter of 1.100 solution in adults, repeating every 5-10 minutes if needed. Adrenaline acts within minutes, increasing blood pressure and reversing bronchospasm. Antihistamines, e.g. clofenamine, block histamine effects, and glucocorticoids, e.g. hydrocortisone, reduce cytokine release and help prevent rebound symptoms. Supportive treatments include nebulized beta-2 agonists, intravenous fluids, and oxygen.
A multi-pronged immediate approach.
Yes. Long-term management involves referral to a specialist to precisely identify the trigger, educate the patient on strict avoidance and episode management, and determine if immunotherapy is indicated. Patients who have experienced anaphylaxis should always be prescribed self-injectable adrenaline epinephrine devices. 2 are recommended.
2 pens, not just 1.
Correct, in case one fails or a second dose is needed. Patients should be instructed on their proper use, advised to carry them always, and to seek immediate medical help after using them. Wearing a medical alert bracelet is also strongly advised. It's worth noting that beta blockers can unfortunately increase the severity of anaphylaxis and reduce the effectiveness of adrenaline.
Beta blockers are a potential problem. Good to know.
Yes, allergy in adolescence warrants special consideration. While many childhood food allergies resolve, allergies to peanuts, tree nuts, fish, and shellfish are often lifelong. This demographic faces the highest risk for fatal food-induced anaphylaxis, often linked to risk-taking behaviors, consuming unsafe foods, denying symptoms, and delaying treatment.
Teenagers. Risk-taking. A dangerous combination with severe allergies.
It can be. The emotional impact can be significant, with some neglecting to carry their auto-injector due to perceived nuisance or social stigma, potentially leading to social isolation.
When the body's defenses aren't just overreacting, but are simply not strong enough, we enter the realm of immune deficiency. This can leave you vulnerable to a host of invaders that a healthy immune system would typically brush aside, creating a constant battle against common pathogens.
This section highlights the critical balance of the immune system. Deficiency can stem from intrinsic genetic defects, which we call primary immunodeficiencies, or from external factors, known as secondary immunodeficiencies. Understanding which component is affected is key to understanding the specific pattern of infection and then, of course, the appropriate management.
Primary versus secondary, born with it versus acquired it.
Basically, yes.
Yeah.
The consequences of immune deficiency are significant and include recurrent infections, paradoxically, autoimmunity due to immune dysregulation, and an increased susceptibility to malignancy, particularly those driven by viral infections like Epstein-Barr virus.
So weakness can lead to infection, autoimmunity, and cancer. Triple threat.
It really underscores the immune system's broad role. While intrinsic defects, those primarily genetic primary immune deficiencies, affect over 100 described conditions and often appear in childhood or adolescence, secondary causes are much more common. These can include infections like HIV and measles, drug therapy, various malignancies, and even the natural process of aging. The clinical presentation truly depends on the specific component of the immune system that is defective, though there's considerable overlap and redundancy within the immune network, making diagnosis sometimes complex.
Okay, let's start with the primary types. What about problems with the phagocytes, the eating cells?
Let's start with primary phagocyte deficiencies. These typically present with recurrent bacterial and fungal infections, often involving unusual sites or forming granulomas. Management requires aggressive treatment of infections, including intravenous antibiotics and surgical drainage, as well as long-term prophylactic antibacterial and antifungal agents.
Constant vigilance against bacteria and fungi.
Yes. Specific examples include chronic granulomatous disease, CGD, where mutations lead to a failure of oxidative killing within phagocytes, making individuals highly susceptible to certain bacterial and fungal infections, particularly catalase-positive organisms like Staph aureus and Aspergillus.
Failure of that respiratory burst we talked about.
Precisely. Another rare example is leukocyte adhesion deficiencies, LADs, where phagocytes cannot properly migrate out of the bloodstream to infection sites, leading to recurrent bacterial infections without pus formation and very high white blood cell counts in the blood because they're trapped in the vessels.
Infections without pus, a key clue.
It is. Defects in the crucial cytokine signaling pathways, like those involving IFN gamma or IL-12, also fall here, making individuals particularly susceptible to mycobacterial infections due to impaired intracellular killing by macrophages.
So problems with the cells themselves or how they move or how they get activated, got it. So those are the cells that literally eat invaders.
Yeah.
What about deficiencies in the complement system? Does that leave people vulnerable to specific types of infections?
Indeed. Complement pathway deficiencies can leave individuals susceptible to particular types of pathogens. While most loss-of-function mutations in complement proteins are rare, a common exception is mannose-binding lectin, MBL deficiency, affecting about 5% of Northern Europeans, many of whom are asymptomatic.
MBL deficiency is common but often silent.
Yes, its clinical significance is debated, but they may have increased bacterial infections if other immune compromises are present. Clinically, patients with deficiencies in the later complement proteins, those forming the ME, present with recurrent infections by encapsulated bacteria, especially Neisseria species, highlighting the importance of the membrane attack complex against these organisms.
The MEC is crucial for Neisseria.
Very much so. Genetic deficiencies of the classical complement pathway also increase the risk of autoimmune diseases, particularly systemic lupus erythematosus, SLE. Interestingly, deficiency of the regulatory protein C1 inhibitor is not associated with recurrent infection, but rather with recurrent angioedema, as we'll discuss later.
C1 inhibitor deficiency causes swelling, not infections.
Correct. Investigations for complement deficiencies typically involve specialized functional tests. If abnormal, individual component measurements follow. Management includes vaccination against common encapsulated bacteria and sometimes lifelong prophylactic penicillin for meningococcal infection.
Moving on to the adaptive system, what happens when there are primary antibody deficiencies? This seems like it would cause a lot of bacterial infections.
You're exactly right. These result from abnormalities in B cell function and are characterized by recurrent bacterial infections, especially of the respiratory and gastrointestinal tract. The most common causative organisms are encapsulated bacteria like Streptococcus pneumonia and Haemophilus influenza.
The usual suspects for respiratory infections.
Yes. These disorders usually present in infancy, after the protective maternal immunoglobulins transferred across the placenta have waned. Key examples include X-linked agamoglobulinemia, a rare X-linked disorder caused by mutations in a gene essential for B cell development, BTK gene, leading to severe bacterial infections in infancy and very low or undetectable antibody levels.
No B cells, basically no antibodies.
Essentially, selective IgA deficiency is the most common primary antibody deficiency, affecting about 1 in 600 Northern Europeans. It's often asymptomatic but can cause recurrent mild respiratory GI infections in about 30% of people. Common variable immune deficiency, CVID, is a heterogeneous adult-onset primary immune deficiency of unknown cause.
CVID, common, variable. Sounds tricky to diagnose.
It can be. It features low serum IgG and a failure to make proper antibody responses, leading to recurrent infections, often bronchiectasis. Paradoxically, CVID is also associated with an increased risk of autoimmune diseases and malignancy, particularly lymphoma.
Weak immunity leading to autoimmunity. That seems counterintuitive.
It highlights the dysregulation aspect. The system isn't just weak, it's out of balance. There's also functional IgG antibody deficiency, a poorly characterized condition marked by defective antibody responses specifically to polysaccharide antigens, like those on encapsulated bacteria. Some used to call this Ig subclass deficiency.
Problems making specific types of antibodies.
Right. Investigations for primary antibody deficiencies typically involve measuring serum immunoglobulins, IgM, IgG, IgA, IgE, B and T lymphocyte subsets, and crucially, specific antibody responses to common pathogens like tetanus and S. pneumonia. If these levels are low, immunization with a killed vaccine is followed by a repeat antibody measurement. Failure to mount a response indicates a significant functional defect.
Checking if they can actually make useful antibodies after vaccination.
Exactly, it's a functional test. Management includes aggressive infection treatment and prophylactic antibiotics. The mainstay for most antibody deficiencies is immunoglobulin replacement therapy, derived from pooled plasma and containing IgG antibodies to a wide range of common organisms.
Giving them the antibodies they can't make.
Precisely. Administered intravenously or subcutaneously, the goal is to maintain trough IgG levels within the normal range to minimize end organ damage. It's often self-administered and lifelong, and importantly, live vaccines should be avoided.
Lifelong treatment, and again, no live vaccines. And what about deficiencies in T lymphocytes? Given their role in orchestrating immunity, these must be quite severe.
They often are. Primary T lymphocyte deficiencies are characterized by recurrent viral, protozoal, and fungal infections. They generally present in childhood and are frequently associated with defective antibody production because T cells are crucial for B cell health.
So T-cell problems hit both cellular and humoral immunity.
Often, yes. Some specific examples. DiGeorge syndrome caused by developmental failure, often a chromosome 22 deletion, that impacts the thymus, leading to very low circulating T-cell numbers. It's also associated with multiple other developmental anomalies like heart defects and hypoparathyroidism. Then there are bare lymphocyte syndromes, rare disorders where HLA molecules are not properly expressed, impairing T-cell development and leading to recurrent infections. Lack of HLA class that can also cause vasculitis due to uncontrolled NK cells.
Problems with the thymus or HLA presentation?
Right. The most severe form is severe combined immune deficiency, SCID. This results from mutations in genes regulating lymphocyte development, leading to a profound failure of T-cell maturation, often with absence of B or NK cells as well.
SCID, the bubble boy disease.
Exactly. The absence of an effective adaptive immune response causes recurrent bacterial, fungal, and viral infections soon after birth. Bone marrow transplantations is the treatment of choice, and gene therapy is an emerging option for certain types of SCID, like ADA deficiency. Finally, autoimmune lymphoproliferative syndrome, ALPS, is a rare disorder caused by a failure of normal lymphocyte program cell death, apoptosis, often due to FAS gene mutations. This leads to a massive accumulation of autoreactive T cells and a range of autoimmune symptoms like anemia and low platelets, plus increased infection risk.
So cells that should die don't, leading to autoimmunity.
Correct. Investigations for T lymphocyte deficiencies primarily involve a total lymphocyte count and quantitation of individual lymphocyte subpopulations, along with serum immunoglobulins. HIV infection should always be tested for in suspected T lymphocyte deficiencies. Management for these patients often includes anti-pneumocystis and antifungal prophylaxis, aggressive infection management, and immunoglobulin replacement for associated antibody production defects. Stem cell transplantation or gene therapy may be appropriate, and antenatal testing followed by early stem cell therapy can significantly improve outcomes if there's a family history.
So many ways the system can be compromised from within, but beyond those primary, often genetic causes, are there more common ways the immune system can be weakened?
Yes, absolutely. We must acknowledge secondary immune deficiencies. These are far more common than primary ones, occurring when the immune system is compromised by external factors.
Things that happen to the immune system.
Exactly. Causes include physiological factors, such as aging, where immune responses decline, immune senescence, reducing vaccine effectiveness and increasing susceptibility to infection. Also, prematurity in pregnancy involves some immune modulation. Infections, like HIV, which directly targets CD4 plus T cells, but also measles and mycobacterial infections, which can suppress immune function. iatrogenic causes, meaning medical interventions such as immunosuppressive therapy for transplants or autoimmune diseases, chemotherapy for cancer, glucocorticoids, radiation injury, or even certain anti-epileptic agents.
Medically induced immune suppression, very common.
Very common. Malignancies, including B-cell malignancies like leukemia, lymphoma, and myeloma, and solid tumors, which can directly suppress immune function or arise from immune cells themselves. Biochemical and nutritional disorders, such as malnutrition, renal insufficiency requiring dialysis, diabetes mellitus, and specific mineral deficiencies like iron and zinc. Other conditions, burns, which disrupt barriers and cause systemic stress, or the absence of the spleen, asplenia, or hyposplenism. Management for these is typically discussed within their respective disease-specific areas, often focusing on treating the underlying condition.
It's clear that immune deficiency presents a major challenge, leaving the body vulnerable. But what about conditions where the immune system isn't underperforming, but rather misfiring or overreacting in a very specific cyclical way? This suggests a different kind of dysregulation entirely.
This takes us into fascinating genetic disorders, where the inflammatory response is aberrantly regulated, often manifesting as periodic fevers. And these chronic, unchecked inflammatory responses can, unfortunately, lead to another serious condition, amyloidosis, where misfolded proteins deposit in organs.
Periodic fevers, like clockwork inflammation.
Sometimes quite predictable, yes.
Yeah.
Periodic fever syndromes are rare genetic disorders characterized by recurrent episodes of fever and organ inflammation, consistently associated with an elevated acute phase response. Let's look at some examples. Familial Mediterranean fever, FMF. This is the most common familial periodic fever, predominantly affecting people of Mediterranean descent. It results from mutations in the MEFV gene, which encodes a protein called pyrin that normally regulates neutrophil-mediated inflammation by suppressing IL-1.
So the brake on inflammation is faulty.
Essentially, yes. FMF is characterized by recurrent painful attacks of fever associated with inflammation of the lining of organs like the abdomen, peritonitis, lungs, pleuritis, and joints, arthritis, lasting a few hours to 4 days, with markedly increased CRP levels. Symptoms completely resolve between episodes, and most individuals experience their first attack before age 20. The major complication of FMF is AA amyloidosis, where protein builds up in organs.
Amyloidosis is a serious long-term risk.
Yes. Management primarily involves colchicine, which significantly reduces febrile episodes in 90% of patients. Mevalonic aciduria, mevalonic kinase deficiency hyper-IgD syndrome. This is an autosomal recessive disorder primarily seen in Western Europe. It's caused by mutations in a gene for an enzyme involved in cholesterol metabolism, though why this causes inflammatory periodic fever remains unclear. It presents with recurrent fever attacks, abdominal pain, diarrhea, lymphadenopathy, and skin lesions. Serum IgD and IgA levels may be persistently elevated. While standard anti-inflammatories are ineffective, IL-1 inhibitors and TNF inhibitors may improve symptoms.
A metabolic problem causing fevers. Interesting link.
The connections aren't always obvious. TNF receptor-associated periodic syndrome, TRAPS, also known as Hibernian fever. This is an autosomal dominant syndrome caused by mutations in a specific gene for a TNF receptor. It presents with recurrent attacks of fever, arthralgia, myalgia, serositis, and rashes that can be prolonged, lasting a week or more. Like FMF, a major complication is amyloidosis, so regular proteinuria screening is advised. Acute episodes respond to systemic glucocorticoids, and IL-1 inhibitors can be effective for prevention.
So several distinct genetic syndromes causing similar patterns of recurrent inflammation, and they can lead to amyloidosis.
These chronic inflammatory conditions can unfortunately lead to amyloidosis, a group of acquired and hereditary disorders characterized by the extracellular deposition of insoluble proteins in various tissues and organs. Pathophysiologically, these deposits consist of specific protein fibrils that misfold and accumulate.
Misfolded proteins gumming up the works.
That's a good description. Deposits can be localized or systemic, and clinical manifestations depend entirely on the affected organs. For example, it might present with nephrotic syndrome affecting the kidneys, cardiomyopathy affecting the heart, or peripheral neuropathy affecting nerves. Diagnosis is established by biopsy of an affected organ, rectum, or subcutaneous fat, with the pathognomonic feature being apple green birefringence under polarized light after Congo red staining.
That green glow is the giveaway.
It is a truly striking visual confirmation. Immunohistochemical staining then identifies the specific protein fibril type. Management aims to support affected organ function and, in acquired amyloidosis, to prevent further deposition by treating the primary cause, which can lead to regression of existing deposits.
Treat the underlying inflammation or conditions, stop the amyloid buildup. Makes sense. It's one thing for the immune system to underperform or overreact to specific external triggers, but what if it mistakes us for an invader? That's the unsettling core of autoimmune disease.
This raises a profoundly important question. How does the immune system, meticulously designed to protect us, turn against our own tissues? The concept of immune tolerance is absolutely key here, and its failure is truly at the heart of these chronic conditions.
Tolerance teaching the immune system friend from foe.
Exactly. Autoimmune disease is defined by immune responses against self-tissue. This can range from harmless, low-titer autoantibodies to significant organ damage and disease. Autoimmune diseases are a major cause of chronic morbidity and disability, affecting up to 1 in 30 adults. The pathophysiology lies in the failure of immune tolerance, the process by which the immune system recognizes and accepts self-tissue.
The friend recognition system breaks down.
Correct. Central immune tolerance occurs during lymphocyte development, where T and B lymphocytes recognizing self-antigens are ideally eliminated before they become fully immunocompetent. However, some autoreactive cells inevitably escape deletion and are then controlled by peripheral tolerance mechanisms. These include suppression by regulatory T cells, functional hyporesponsiveness, anergy, and lymphocytes encountering antigen without co-stimulatory signals, and programmed cell death, apoptosis. Autoimmune diseases develop when self-reactive lymphocytes evade these crucial tolerance mechanisms.
So multiple checkpoints, central and peripheral, need to fail.
It's usually a multi-hit process. Multiple contributing factors play a role. Genetic factors are significant. Autoimmune diseases are more common in women, and many are associated with genetic variations in HLA loci, reflecting HLA's importance in shaping lymphocyte responses. Other susceptibility genes have also been identified, like PTPN22, CTLA4, IL-23R. However, despite strong genetic associations, these have limited predictive value for individuals.
Genes increase risk but don't guarantee disease.
Precisely. Environmental factors also contribute, with infection being the most widely studied, alongside cigarette smoking and hormone levels. Infection-triggered autoimmunity can occur through several mechanisms. Molecular mimicry, where there's cross-reactivity between pathogen and host proteins, like Guillain-Barré after Campylobacter infection. The release of sequestered antigens from damaged tissues that are usually hidden from the immune system, and the production of inflammatory cytokines that overwhelm normal control mechanisms, causing bystander damage. Occasionally, drug-induced autoimmunity can occur.
So genes load the gun, environment pulls the trigger.
Often seems that way, yes. The clinical features of autoimmune diseases are highly variable, classified by organ involvement or the predominant mechanism of tissue damage. The widely used Gell-Coombs classification of hypersensitivity distinguishes 4 types of immune responses causing tissue damage. Type 1 is allergy, not directly autoimmune.
Right, Gell-Coombs. Types 2, 3, and 4 are relevant here.
Yes. Type 2, antibody-mediated hypersensitivity, involves cytotoxic antibodies to GI-GM, binding to cell surface antigens, causing cell killing, as seen in autoimmune hemolytic anemia or Goodpasture's disease. Type 3, immune complex-mediated hypersensitivity, result from antibodies binding to soluble antigens to form immune complexes, which then deposit in tissues and trigger inflammation via complement and phagocytes. This leads to systemic diseases like systemic lupus erythematosus, SLE, or cryoglobulinemia. Type 4, delayed type hypersensitivity, is mediated by activated T cells and macrophages that together cause tissue damage, as seen in type 1 diabetes and Hashimoto's thyroiditis.
Different mechanisms lead to different diseases. Makes sense. How are these investigated?
For investigations, autoantibodies are crucial for diagnosis and monitoring. They can be quantified or qualitatively assessed, like antinuclear antibodies, ANA, which show different staining patterns. Complement measurements are also useful in immune complex-mediated diseases. Type 3. Classical pathway activation leads to decreased C4, often C3, and serial levels can be a surrogate for disease activity, as in SLE. Cryoglobulins are antibodies against other immunoglobulins that precipitate in the cold, causing type 3 hypersensitivity reactions like rash, arthralgia, neuropathy. Testing requires careful sample handling at body temperature.
Measuring the weapons, autoantibodies, and the collateral damage, complement consumption.
A good summary. Management depends on the affected organ system, generally involving glucocorticoids and immunosuppressive agents, increasingly combined with biologics targeting specific cytokines or receptors. However, not all conditions require immune suppression. For example, celiac disease is managed by gluten withdrawal and autoimmune hypothyroidism with thyroxine replacement.
Building on the idea of immune misfires, allergies represent a massive slice of chronic illness affecting so many people. It's the immune system, designed to protect us, going into overdrive against things that are completely harmless, things like pollen, peanuts, or even a bee sting, turning them into perceived threats.
This is a perfect illustration of the hygiene hypothesis at play, where our modern environment might ironically contribute to this widespread immune oversensitivity. The key is understanding how a seemingly harmless substance can trigger such a dramatic IgE-mediated response.
IgE again, like in anaphylaxis.
Exactly. Type I hypersensitivity is the classic allergic reaction mediated by IgE. Allergy overview. Allergies are common and increasing, affecting 15–20% of the population. Atopy is the genetic predisposition or tendency to produce an exaggerated IgE immune response to otherwise harmless environmental substances. Allergic disease is the clinical manifestation of this inappropriate IgE immune response.
Atopy is the tendency, allergy is the disease.
Correct. In terms of pathophysiology, normally the immune system tolerates environmental substances. However, in allergic individuals, initial exposure to an allergen triggers the production of specific IgE antibodies by activated B cells. These IgE antibodies then bind to high-affinity IgE receptors on mast cells, a process called sensitization, which doesn't cause symptoms yet.
Loading the mast cells with IgE triggers.
Precisely. Upon re-exposure to the allergen binds to and cross-links these IgE-coated mast cells, activating them and causing the rapid release of vasoactive mediators, leading to the early phase response or type I hypersensitivity. This can manifest as immediate symptoms like sneezing, rhinorrhea, urticaria, angioedema, wheezing, and in severe cases, anaphylaxis.
The immediate reaction we typically think of as allergy.
Yes. In some individuals, this is followed by a late-phase reaction, characterized by persistent mast cell activation, ongoing swelling, and local inflammation, mediated by mast cell metabolites, basophils, and eosinophils. Long-standing or recurrent allergic inflammation can lead to chronic allergic inflammation, where a complex infiltrate of various immune cells is present, and antihistamines alone become ineffective.
So it can become a chronic smoldering inflammation too.
It can. The rising incidence of allergic diseases is not fully explained, but the hygiene hypothesis is a widely held theory. It proposes that early life infections are critically important for immune maturation, biasing the immune system against developing allergies. Thus, the high prevalence of allergic disease in developed countries is seen as a penalty for decreased exposure to infections due to improved sanitation and healthcare.
Too clean for our own good, immunologically speaking.
That's the essence of the hypothesis, though it's likely more complex than just cleanliness. Genetic factors also contribute strongly, with a positive family history being common, and specific gene variants influencing susceptibility.
Nature and nurture both playing a role.
Definitely. Clinical features of allergy can affect various systems: dermatological, urticaria, atopic eczema, angioedema; respiratory asthma, atopic rhinitis; ophthalmological, allergic conjunctivitis; gastrointestinal, food allergy; and other anaphylaxis, drug allergy, insect venom allergy.
Affecting skin, lungs, eyes, gut, pretty much everywhere.
It can manifest in many ways. Let's touch on some specific allergies. Insect venom allergy can lead to severe reactions, but antigen-specific immunotherapy, desensitization, can significantly reduce recurrent anaphylaxis risk from 50–60% down to about 10%. Peanut allergy is the most common food-related allergy, often presenting early in life and carrying a high risk of accidental exposure due to its ubiquity. Birch oral allergy syndrome links birch pollen hay fever with oral symptoms, itching, swelling, after eating certain raw fruits, vegetables, or nuts due to cross-reactive allergens that are destroyed by cooking.
Interesting cross-reactivity there between pollen and food.
It's quite common. For diagnosis, it's crucial to differentiate true IgE-mediated hypersensitivity from other reactions. We establish symptoms, specific triggers, predictability, and time lag. Skin prick tests are a key investigation. A droplet of diluted allergen is placed on the forearm and superficially punctured, with a positive response showing a local wheal and flare.
Quick visual confirmation.
Yes, and patients see the result, which helps adherence. Specific IgE tests, blood tests, are an alternative, quantifying IgE against a suspected allergen, useful when skin testing is inappropriate. Supervised allergen exposure, allergen challenge, is performed in specialist centers for carefully selected patients, providing definitive diagnosis, especially for food, drug, or occupational allergies. Mast cell tryptase measurement is extremely useful for investigating possible anaphylaxis. Serum total IgE measurements are not routinely indicated. as levels can be high for many reasons, like parasites, and normal levels do not exclude allergic disease.
So skin tests and specific IgE are the mainstays for identifying triggers.
Generally, yes. Management of allergic individuals involves several approaches. Avoidance of the allergen is rigorously attempted in all cases. Antihistamines inhibit histamine effects and are useful for prophylaxis, especially long-acting, non-sedating ones. Glucocorticoids are highly effective, often used topically, nasal sprays, inhalers, creams, to minimize adverse effects. Sodium chromoglicate stabilizes mast cell membranes, inhibiting mediator release, primarily for prophylaxis in asthma or rhinitis. Antigen-specific immunotherapy, desensitization, involves sequential administration of increasing allergen extract doses over a prolonged period, often years. It's highly effective for preventing insect venom anaphylaxis and grass pollen allergic rhinitis, now increasingly given sublingually. Omalizumab, a monoclonal antibody against IgE, inhibits IgE binding to mast cells and basophils, licensed for refractory chronic spontaneous urticaria and severe allergic asthma. And of course, adrenaline, epinephrine, administered via preloaded self-injectable devices, is life-saving in acute anaphylaxis.
Angioedema, a localized, often dramatic swelling, frequently looks alarming. When we see this, is it just a type of allergy, or is there a distinct underlying mechanism at play here that differentiates it? It often occurs with hives, urticaria, but sometimes without. Right.
That's a key point. This raises an important question. While it can certainly be a manifestation of allergy, angioedema also frequently stems from entirely different biochemical pathways, particularly those involving bradykinin rather than histamine. Differentiating these mechanisms is absolutely crucial for effective treatment.
Histamine versus bradykinin, different chemicals causing the swelling.
Exactly. Angioedema is defined as episodic, localized, non-pitting swelling of submucous or subcutaneous tissues. Its pathophysiology and types are varied. It can be an allergic reaction to a specific trigger. This is IgE-mediated mast cell degranulation. Main mediator histamine, common, usually occurs with urticaria and often a history of other allergies, idiopathic angioedema. Also common, involves non-IgE-mediated mast cell degranulation, main mediator histamine, usually occurs with urticaria, sometimes physical triggers, heat, pressure, exercise.
So those two types are histamine-driven and often have hives. What about the others?
Then we have two important bradykinin-mediated types, hereditary angioedema, HAE, inherited C1 inhibitor deficiency. This is a rare genetic disorder caused by decreased production or activity of the C1 inhibitor protein. Its deficiency leads to increased local bradykinin release, which is the key mediator, causing pain and swelling. Crucially, this type occurs without urticaria. ACE inhibitor-associated angioedema occurs in a small percentage of patients taking ACE inhibitor drugs for blood pressure heart failure. The drug inhibits the breakdown of bradykinin, leading to its accumulation and swelling. Again, this typically occurs without urticaria.
So HAE and ACE inhibitor swelling are bradykinin-driven and usually lack hives. That's a critical distinction.
Absolutely critical for diagnosis and treatment. The clinical features include soft tissue swelling, most frequently affecting the face, but also extremities and genitalia. Life-threatening complications arise from laryngeal or tongue involvement, causing respiratory tract obstruction. GI involvement, where there's edema of the intestinal mucosa, leads to severe abdominal pain and distension, often leading to unnecessary exploratory surgeries in undiagnosed HAE patients.
Swelling inside the gut causing severe pain. Wow.
Yes. HAE specifics. Episodes can be spontaneous or triggered by local trauma or infection, and they're self-limiting, typically resolving within 48 hours. Patients usually present in adolescence but can go undiagnosed for years. A family history is present in 80% of cases. As mentioned, it's not associated with allergic diseases or urticaria. ACE inhibitor angioedema typically affects the head and neck, can occur years after starting the drug, and is also not associated with urticaria.
How do you investigate this? That C1 inhibitor seems key for HAE.
Investigations are critical for differentiating the mechanism. A clinical history of allergy or drug exposure provides important clues. If no obvious trigger, especially if urticaria is absent, measuring complement C4 is useful. A low C4 level suggests hereditary or acquired angioedema, HAE. If C4 is low, further investigations are pursued to look for C1 inhibitor deficiency, both levels and function.
Low C4 points towards C1 inhibitor problems.
This is a good screening test. Management depends entirely on the underlying cause. Allergic idiopathic angioedema, histamine-driven, generally responds to antihistamines and glucocorticoids. For drug-induced ACE inhibitor angioedema, drug withdrawal is necessary, though swelling can persist for a limited time afterwards. Rechallenge is contraindicated, and ARBs, another class of BP meds, should be used cautiously. Hereditary angioedema, HAE, and acquired C1 inhibitor deficiency, braiding and driven, are unresponsive to antihistamines and glucocorticoids.
Standard allergy treatments won't work for HAE.
Correct. Acute attacks are treated with specific therapies like purified C1 inhibitor concentrate or a bradykinin receptor antagonist, a catabant. Prevention involves agents like anabolic steroids, danazol, or tranexamic acid. Patients can be taught self-administration, and medical alert is advised. Acquired C1 inhibitor deficiency is clinically indistinguishable from HAE, but presents in late adulthood, often due to underlying autoimmune or lymphoproliferative diseases producing antibodies against C1 inhibitor.
Transplantation is truly a medical miracle, offering a new lease on life to those with end-stage organ disease. But for the immune system, a transplanted organ is, quite simply, a foreign invader. This sets up a complex and potentially devastating battle within the recipient's body.
This raises an important question. How do we harness the immune system's power to accept a life-saving transplant, while simultaneously preventing it from destroying that very graft? It's a delicate balance of suppression and vigilance, a constant immunological tightrope walk.
Keeping the immune system quiet enough, but not too quiet.
Exactly. Transplantation serves as a definitive treatment for end-stage organ disease. However, it comes with major complications. Graft rejection, drug toxicity from immunosuppressants, and infection due to the necessary immunosuppression. Transplant survival has significantly improved due to less toxic immunosuppressive agents and a deeper understanding of rejection processes.
We've gotten better at managing that tightrope.
We have. Transplant rejection is an inevitable immune response by the recipient unless the transplant is between monozygotic twins. The type and severity of rejection are determined by the genetic disparity between donor and recipient, primarily HLA proteins, which are highly polymorphic and almost always recognized as foreign, the immune status of the host, and the nature of the transplanted tissue.
HLA matching is key.
Very key. There are several types of rejection. Hyperacute rejection occurs minutes to hours post-transplant, leading to thrombosis and necrosis. It's mediated by preformed recipient antibodies against donor HLA antigens, type 2 hypersensitivity. This is very rare in clinical practice today due to rigorous pre-transplant screening and cross-matching.
We test for those preformed antibodies now.
Yes, it's standard practice.
Yeah.
Acute cellular rejection. The most common form, occurring 5-30 days post-transplant, characterized by cellular infiltration, T cells attacking the graft type 5V hypersensitivity. It causes graft dysfunction, pain, tenderness, worsening function, and is usually amenable to increased immunosuppression. Acute vascular rejection, or antibody-mediated rejection, also occurs 5-30 days post-transplant, involving inflammation of blood vessels, vasculitis, mediated by antibodies formed de novo after transplantation. It reduces graft survival and may require aggressive immunosuppression, sometimes even plasmapheresis to remove the antibodies.
So rejection can be cell-based or antibody-based, even early on.
Correct. And not all anti-donor antibodies cause damage. It depends on their type and ability to activate things like complement. Chronic allograft failure, chronic rejection, occurs more than 30 days post-transplant and is a major cause of long-term graft loss. Pathology shows fibrosis and scarring within the organ and proliferation of vascular smooth muscle narrowing blood vessels. Its pathogenesis is poorly understood but involves a combination of immunological damage, sudden rejection, non-immune factors like hypertension and drug toxicity.
A slow, smoldering damage over time.
Unfortunately, yes. Investigations for transplantation are extensive. Pre-transplantation testing includes HLA typing to determine donor-recipient compatibility and screen for pre-existing anti-HLA antibodies in the recipient. Donor-recipient cross-matching is a crucial functional assay performed immediately before transplant, testing if recipient serum can bind to and kill donor lymphocytes. A positive cross-match contraindicates transplantation due to the high risk of hyperacute rejection. Post-transplant biopsy with C4D staining is important. C4 deposition in graft capillaries indicates antibody-mediated damage via the classical complement pathway, aiding early diagnosis of vascular rejection.
C4 staining shows complement activation in the graft.
Yes, a sign of antibody attack. Complications of transplant immunosuppression are significant. Transplant recipients require indefinite treatment, typically with 2 or more synergistic immunosuppressive drugs to minimize adverse effects. The major complications are infection and malignancy.
The price of suppressing rejection.
It is. Opportunistic infections are minimized by prophylaxis, for example, antivirals for CMV, antibiotics for pneumocystis. Immunization with killed vaccines is appropriate, but live vaccines should never be given. Malignancy risk increases because T-cell suppression impairs control over viral infections linked to malignant transformation. like EBV causing lymphoma, HHV8 causing Kaposi sarcoma, HPV causing skin tumors, and also slightly increases the risk of common non-viral cancers, highlighting the role of T cells in anti-cancer surveillance. Various immunosuppressive drugs are used, like antiproliferatives, azathioprine, mycophenolate, calcineurin inhibitors, cyclosporin, tacrolimus, glucocorticoids, antibody therapies, ATG, baseliximab, and newer agents like belatacept,
A whole arsenal of drugs to dampen the immune response.
each targeting different aspects. A major problem in transplantation remains the shortage of organ donors. Cadaveric donors, brainstem death, are insufficient to meet current needs. Living donors are an alternative, especially for renal transplantation, with altruistic donation, usually from close relatives, being common. Living donation carries risks to the donor and is highly regulated. Non-altruistic organ donation, organ sale, is illegal in most countries due to ethical concerns about coercion and exploitation.
Finally, let's explore one of the most exciting and rapidly evolving areas in immunology, the immune system's role in fighting cancer. For a long time, we knew our bodies fought off invaders, but could it be our ultimate weapon against this disease, even recognizing and eliminating our own rogue cells?
This raises an important question. While the immune system can recognize and eliminate cancer cells, tumors have evolved remarkable strategies to evade these defenses. Understanding these evasion mechanisms has been the key to unlocking powerful new therapies.
So the immune system tries to fight cancer, but cancer fights back.
Exactly. Immune surveillance is critically important for monitoring and removing damaged or mutated cells as they arise. However, the ability of the immune system to effectively kill cancer cells is influenced by the tumor's immunogenicity, how recognizable it is to the immune system. Tumors employ various tumor evasion mechanisms. Many cancer antigens are poorly expressed, or specific antigens can mutate, allowing the tumor to evade immune responses. Furthermore, tumors can co-opt the very inhibitory pathways that the immune system normally uses to maintain self-tolerance, effectively turning the body's own checks and balances against itself.
Using the immune system's own brakes to protect themselves. Clever, but devastating.
It is. The recognition and understanding of these immune checkpoint pathways have led to the development of a number of groundbreaking therapies for cancers that were previously refractory to treatment. These therapies work by essentially releasing the brakes on the immune system, allowing T cells to attack cancer.
Checkpoint inhibitors.
Precisely. For example, antibodies targeting CTLA4, one of those T cell brakes we mentioned, are licensed for refractory melanoma, and antibodies targeting PD-1, programmed cell death protein 1, or its ligand PDL1, are now used in various cancers like melanoma, non-small cell lung cancer, and renal carcinoma, often with dramatic results.
Unleashing the T cells against the tumor.
That's the goal. However, a crucial point to remember is the potential risk. These therapies can sometimes lead to the development of autoimmunity, causing inflammatory side effects in various organs. That precisely reflects the vital importance of these very pathways in controlling self-tolerance and preventing the immune system from attacking healthy tissues in the first place.
Releasing the brakes on T cells can sometimes lead to them attacking normal tissue too. Another tightrope.
It is indeed. Managing these immune-related adverse events is a key part of using these powerful therapies effectively. #tag outro.
Wow. What an incredible deep dive into the human immune system. From those initial physical barriers that keep pathogens out, to the incredibly sophisticated cellular and molecular machinery that orchestrates defense, response, and repair, it's abundantly clear our body's defense system is astonishingly complex and powerful.
Indeed. It's a constant dynamic balance, a finely tuned system. And what's truly remarkable is how a system designed for protection can, when dysregulated, lead to such diverse and debilitating diseases, from chronic inflammation and autoimmune conditions to severe allergies and vulnerabilities to infection.
Yeah.
But equally remarkable is how our growing understanding allows us to precisely manipulate it for therapeutic benefit, treating diseases that were once untreatable.
It makes you appreciate the incredible resilience of the human body, operating on so many levels, usually seamlessly. But it also highlights how vulnerable we can become when that delicate balance is disturbed. For you, our listener, this deep dive should leave you with a profound appreciation for the silent war being waged inside us every single day, keeping us alive and well.
Consider this. The immune system's profound ability to learn and adapt has driven its evolution over millennia, constantly refining its strategies against an ever-changing array of threats. As we continue to unravel its deepest secrets, and our capacity to engineer immune responses becomes increasingly sophisticated. Think SAR T cells, mRNA vaccines, targeted biologics. How might this fundamentally change the very definition of human health and disease in the coming decades?
That's a powerful question to ponder. Where does manipulating immunity take us? We hope this deep dive has sparked your own curiosity and armed you with crucial insights into this fascinating field. We encourage you to continue your own deep dives into specific areas of interest that resonated with you today. Until next time, stay curious.